Now we start our lecture. We were discussing about the reproductive system, especially of male. Towards the lumen of seminiferous tissues are layer of progressively more progressively more mature cells. In order to advancing maturity, these are the primary spermatocytes, then secondary spermatocytes, then spermatocytes, sperm- spermatides, and then sperm cells are formed. This is the process which takes place. After a sperm or spermatozoa has formed, it is released into the lumen of the seminiferous tubules. The plural terms are sperms and spermatozoid, who are embedded among the spermatogenic cells in the seminiferous tubules are large Sertoli cells or sublenticular cells, which extend from the basement membrane to the lumen of the tubule. Internal to the basement membrane and spermatogonia, tight junction point neighboring Sertoli cells to one another. These junctions form an obstruction known as the blood-testis barrier because substances must first pass through the Sertoli cells before they can reach the developing cells. By isolating the developing gametes from the blood testis barrier prevent an immune response against the spermatogenic cells is surface antigens, which are recognized as a foreign by the immune system. So the blood testis barriers, they protect the spermatogonia because the immune system take these spermatozoa as an antigen outside as an outsider and create an antigen. So the blood testis barrier does not include spermatogonia. In the spaces between the adjacent seminiferous tubules are the cluster of cells called the Leydig interstitial cells. These cells secrete testosterone, which is a hormone, especially male hormone, the most prevalent androgen. An androgen is a hormone that promotes the development of masculine characteristics. The characteristic of the male that's like the voice, mustaches, and the armpit hairs. These are all representation of a muscular characteristic. Testosterone are also promote a man's libido sexual drive. As much as the testosterone is present in a male, he has got more desire for the sex. Spermatogenesis. In human, spermatogenesis takes 65 to 75 days, means sperms are produced and prepared, go into the process, and it will take about 65 to 75 days. It begins with the spermatogonia, which contains the diploid 2n number of the chromosome. When spermatogonia are type of the stem cells which they undergo mitosis division. Some spermatogonia remain near the basement membrane of the seminiferous tubules in an undifferentiated state to serve as a reservoir of cells for future division and subsequent sperm production. The rest of the spermatogonia lose contact with the basement membrane, squeeze through the tight junction of the blood testis barrier, undergoes developmental changes and differentiate into primary spermatozoites. Primary spermatocytes like spermatogonia are didiploid 2n, that is, they have 46 chromosomes. Shortly after it forms, each primary spermatocytes replicate its DNA and then mitosis begin in meiosis begin. In meiosis, one homologous pair of the chromosomes line up at the metaphase plate and crossover occurs. Then the meiotic spindle pulls one duplicated chromosomes of each pair to an opposite pole of the dividing cells. The two cells formed by the meiosis one are called the secondary spermatocytes. Each secondary spermatocyte has a 23 chromosomes. The, the haploid number and each chromosome within a secondary spermatocyte, however, is made up of two chromatids, two copies of the DNA still attached by the centromere. No replication of the DNA occurs in the secondary spermatocytes. In meiosis II, the chromosomes line up in a single file along the metaphase plate and the two chromatids of each chromosome separates. The four haploid cells resulting from the meiosis II are called spermatid. A single primary spermatocyte therefore produces four spermatid via two rounds of the cell's division, meiosis I and meiosis II. So this is the process, how the whole process takes place for the development of sperms. And that is the male from the male gamete. A unique process occurs during spermatogenesis. Genesis means to produce sperms. Spermatogenic cells proliferate. They fail to complete cytoplasmic separation, cytokinesis. The cell remain in contact with cytoplasmic bridges through their entire development. This pattern of development most likely account for the synchronized production of sperm in any given area of the seminiferous tubules. So sperms are produced in seminiferous tubules, produced in the testes, and then it will move to the seminiferous tubules, and then it will take a shape of sperms. It may also have a survival value in that half the sperms contain an X chromosome and half contain a Y chromosome. This is for male, because in female there are X and X chromosomes. The target X chromosomes may carry genes needed for spermatogenesis that are lacking on the smaller Y chromosomes. The final stage of spermatogenesis is the development of diploid spermatid into sperms. No cell division occurs in spermatogenesis. Each spermatid transforms into elongated cylindrical sperm. Means now the sperms will take the shape of the head, body, and tail. The head and acrosome, described shortly, form atop the nucleus, which condense and elongated. A flagellum development. Flagellum means tail, which is movable, and that's why the sperm in semen moves from one place to another place. And mitochondria multiply, means the energy packets also multiply to provide energy for the division and development. Satori cells dispose of the excess cytoplasm that sloughed off. So this is the whole procedure of the spermatogenesis, as you have seen. So further, if you will go, finally, sperms are released from their connection to Satori cells, and event known as spermation. Sperms then enter the lumen of the seminiferous tubules Fluid secreted by the Sertoli cells pushes the sperm along their way to the duct of the testis. At this point, sperms are not yet able to swim because here there is one liquid is secreted from the testis in which they swim. Sperm. Each day, about 300 million sperms complete the process of spermatogenesis. A sperm is about 60 meters long and contains several structures that are highly adaptable for reaching the penetrating a secondary oocyte. The major part of the sperms are the head and tail. The flattened pointed head on the sperm is about 4 to 5. This is make a correction, I think. This is millimeter long. It contains a nucleus with 23 highly condensed chromosomes. Covering the interior two-thirds of the nucleus is the acromes, means head, a cap-like vesicles filled with the enzyme that help the sperms to penetrate a secondary oocyte, means in the ovum, to bring about a fertilization, means zygote. Among the enzymes are hyaluronidase and proteases. The tail of the sperm is subdivided into four parts: neck, middle piece, principal piece, and end piece. The neck is the constricted region just behind the head that contains centrioles. The centrioles form the microtubules that comprise the remainder of the tail. The middle piece contains mitochondria arranged in a spiral, which provides the energy ATP adenotrophosphates for locomotion, for movement of sperms to the site of fertilization, and for sperm's metabolism. The principal piece is the longest portion of tail, and the end piece is the terminal tapering portion of the tail. Once ejaculated, most sperms do not survive with more than 48 hours within the female reproductive system. Hormonal control of these all processes of the testis, although the initiating factors are unknown at the puberty, certain hypothalamic neurosecretory cells increase their secretion of gonadotropic releasing hormone GnRH. The hormone is turn stimulated gonadotropin in the anterior pituitary to increase their secretion of two gonadotropic tropics, luteinizing hormone and FSH follicular stimulating hormone. The pituitary secretes about 9 hormones, 7 from the anterior lobe and 2 from the posterior lobe. Oxytocin and vasopressin are secreted from the posterior lobe, and from anterior lobe there are 7 hormones secreted. One is the gonadotropic hormone, which further make 2 more hormones, which is luteinizing hormone and follicular stimulating hormone. Means follicles means ovums, which develop from the follicles, and then luteinizing hormone. Show the hormone and negative feedback loops that control secretion of the testosterone and spermatogenesis. Luteinizing hormone stimulates Leydig cells, which is a precursor of the sperms, which are located between the seminiferous tubules to secrete the hormone testosterone. This steroids hormones is synthesized from the cholesterol in the testes, means fat in the testes, and is the principal androgen. It is lipid soluble and readily diffuses out of the Leydig cells into the interstitial fluid and then into the blood. Via negative feedback secretions, Why negative feedback testosterone suppressed secretion of LH by anterior pituitary gonadotropin and suppressed secretion of GnRH by hypothalamic neurosecretory cells. In some target cells, such as those in the external genital and prostate, the enzyme 5-alpha reductase convert testosterone to the another androgen called the dihydrotestosterone. So in this way, the hormonal control is there for this whole process. FSH, follicular stimulating hormone, acts indirectly to stimulate the spermatogenesis. FSH and testosterone acts synergistically on the Sertoli cells to stimulate secretion of androgen binding proteins ABP into the lumen of seminiferous tubules and into the interstitial fluid around the spermatogenesis genetic cell. Androgen binding protein ABP binds to testosterone. Keeping its concentration high, testosterone stimulates the final step of spermatogenesis in the seminiferous tubules. It means in the seminiferous tubules, sperms are produced with the help of testosterone. Once the degree of spermatogenesis required for male reproductive function has been achieved, Sertoli cells release inhibin, a protein hormone named for its role in the inhibiting follicular FSH, follicular stimulating hormone secretion by the anterior pituitary. So negative feedback mechanism means when there is no need of spermatogenesis, then inhibin enzyme hormone is released, a protein hormone is released, and it will inhibit the anterior pituitary function for production of FSH, follicular stimulating hormone. If spermatogenesis is proceeding too slowly, less inhibin is released, which permit more FSH secretion and increase rate of spermatogenesis. So sperms are developed. They are also known as follicles. Female follicles stimulating hormone produce the ovums, and in male sperms. And it will help in the generation of sperms is known as spermatogenesis. Testosterone and dihydrotestosterone both bind to the same androgen receptors which are found within the nucleus of the target cell. The hormone receptor complex regulates genes expression, turning some genes on and other off. Because of these changes, the androgen produces several effects. So now you must have understand some of the actions which is taking place in different places in the development of sperms and the role of the pituitary gland, and the pituitary gland produces follicular stimulating hormone and luteinizing hormone, as well as in Sartori cells, the testosterone will convert into dihydrotestosterone. So we have, it's a complete process how these cells are produced and what is the role of these cells and how the male reproductive system works. So this is all about today lecture. Then we discuss about the prenatal development at the time of the birth, how this whole process takes place in a child, and at the time of the maturity, the sperms are also matured and it will come out with the ejaculation. And the role of pituitary and other hormones is also there for controlling purpose. So this is all about today's lecture, and we will see you in the next lecture. Thank you.